Hello, welcome to the Aspirational Neuroscience Journal Club. This week we are covering this 2015 Nature paper, Selective Corticostriatal Plasticity During Acquisition of an Auditory Discrimination Task. This is a truly fantastic paper that tests our theories of how reinforcement learning is physically encoded in the rat brain. The key result is that the authors could perfectly determine, on a rat-by-rat basis, the behavioral task each rat learned solely by measuring the pattern of strengthened synaptic connections in the rat's striatum. These measurements were made after each rat was dead, by careful electrophysiological measurements of an acute slice of the rat's brain. Here's a selective quote from the paper. Thus, postmortem study of corticostriatal efficacy can reliably reveal the training history of individual subjects. This amazing result has actually been replicated now and extended in a following 2021 paper shown here. Here's a selected quote from that paper. The sign of this association can be read out post-mortem with nearly perfect fidelity using electrophysiological measurements from a single acute brain slice. Let's jump right into the details. The authors employed a tone discrimination 2 alternative force choice task to study the physical basis of reinforcement learning in the brain. For example, rats had to learn to lick at a liquid port on the right when they heard a high frequency tone, and lick at a left port when they heard a low frequency tone. The task is referred to as the low right task. Other rats were trained with the opposite low left task. The auditory stimuli used were cloud of tones stimuli, which are complex enough to activate a range of neurons in auditory cortex. Many previous experiments have shown that the striatum, the input node of the basal ganglia, is central to reinforcement learning. The striatum receives axonal inputs from all over the cortex. Striatal spiny neurons project to motor circuits that release behaviors. And dopamine reward signals innervate the striatum, guiding its learning. Other studies have shown that corticostriatal synapses, like the ones that are studied in the current paper, physically enlarge when strengthened via an LTP glutamate uncaging protocol, but only when dopamine is present. This large body of research led to the current author's study of these synapses of auditory cortical axons onto striatal neurons as being the likely site of the learning in this task. In their first experiment, the authors developed a method to record synaptic changes in vivo. Authors injected the auditory cortex of a rat with an AAV virus expressing the optogenetic channel rhodopsin 2. This means that they could shine light pulses into specific areas of the striatum where the infected auditory cortex axons projected, and cause those axons to fire, releasing their glutamate on striatal dendrites in that illuminated region. Using an implanted electrode, they measured the local field potential response to these optogenetic light pulses. They found that this local field potential response could be interpreted to measure the strengthening of synapses due to learning. The next slides show how this works. Let's zoom in on a tiny piece of the striatum, showing the synapse of a single axon from auditory cortex onto a striatal dendritic spine. Here's the axon from the auditory cortex. Here is that striatal neuron's dendritic tree. There are synaptic vesicles filled with glutamate ready to be released if the axon fires. And there are AMPA receptors on the postsynaptic spine that will allow positive currents to flow into that cell when glutamate is bound to them. And here are the channel rhodopsin 2 channels that will allow the experimenters to fire that axon whenever they want to via a light pulse. Let's walk through an experiment. A light pulse is activated, causing positive sodium ions to enter the axon. This shows up as a large negative transient in the local field potential recordings. The action potential created in the axon causes release of glutamate at its terminals, which then binds to AMPA receptors in the postsynaptic membrane. This generates more positive currents, this time into the striatal neuron, which shows up as a later negative transient in the local field potential recording. The authors argue that the initial slope of this late component is proportional to the summed synaptic strength in that region. A small synapse will have few AMPA receptors and will thus show a small transient, like this local field potential recording highlighted in red here. The slope of this will be shallow. In contrast, a larger synapse will have more AMPA receptors as shown here. It will show a larger transient like that highlighted in red here, and this will have a steeper slope. I want to stress that this is, of course, an oversimplification. The late component of this local field response to an optogenetic pulse is an average over thousands of synapses in the region, along with many other downstream effects. But this is what the authors argue can measure the overall strengthening of auditory cortical striatal synapses in a region. The authors do a range of controls to verify this interpretation that I won't cover here, but these controls include measuring the effect after applying chemical blockers and after changing ionic concentration ratios. Having developed this method to record corticostriatal synaptic strength in vivo, the authors then trained rats in a low right task and recorded optical local field potential responses in the left striatum. Let's walk through the logic of this experiment. Here's a quote from the paper. All animals in this cohort were trained to associate low-frequency sounds with rightward choices, and all recordings were performed in the left striatum. Since the left striatum invokes contralateral licks, it would presumably be the one whose synapses would be strengthened to learn to evoke a rightward lick to a low tone. So the hypothesis is that synapses from low-frequency auditory cortical neurons to left striatal neurons would be strengthened when learning this task. In contrast, synapses from high-frequency neurons would not be strengthened in the left striatum at all. But how can one isolate only the low-frequency axons? It turns out that some areas of the striatum receive mainly low-frequency axons and others mainly high-frequency axons. In each rat, the authors first performed an auditory measurement to determine the frequency tuning of that region that their electrodes happened to be implanted in. Then they recorded how this channel rhodopsin 2 evoked local field potential slopes changed over days of learning. Here are the recordings from one rat during training. The electrodes in this rat happen to be implanted in a region of the striatum receiving low-frequency axons. So the prediction is that the synapses would show strengthening over the course of the training. And indeed, that's exactly what the authors found. You can see in this curve that the channel rhodopsin 2 local field potential slope starts off without learning at a level like this, and then over days of training sessions it increases. And this is the plots of that local field potential changing over those different training days. In contrast, here are the recordings from a rat whose electrodes were implanted in a high-frequency region of the left striatum. It shows no changes in synaptic strength over training, again, just as theory predicted. And here are the results for all rats in this particular experiment. It shows that synapses of auditory cortical axons responding only to low frequencies onto the left striatal neurons were strengthened during learning, just as predicted by theory. The authors also did a vision-based control experiment to show that the effects that they were seeing were not just due to general motor learning in the striatum. But I won't cover that here. In their final experiment, the authors developed an in vitro brain slice preparation so that the previous experiment could be done in several known tonotopically mapped regions of the striatum of a single rat. This was important to allow robust, accurate measurement of the relative learned synaptic strengthening within a single rat. The authors mapped general tonotopy of striatal inputs by injecting two different color tracers. A green fluorescent virus was injected into the caudal part of the auditory cortex, which processes low frequencies, and a red fluorescent virus was injected into the rostral part of the auditory cortex, which processes high frequencies. This allowed them to verify that the striatum itself has a tonotopic axis. Reading from the paper here, cortical axons terminated in the striatum in distinct bands, with cortical projections tuned to high-frequency sounds terminating more laterally in the auditory striatum, and projections tuned to low-frequency sounds more medially. With this knowledge, the authors decided to do all further experiments on acute brain slices, one taken from each rat that had been trained on a particular task while they were alive. Each rat was sacrificed so that their brain could be quickly sliced and a single 350 micron slice through the left striatum retrieved for acute slice electrophysiology. The acute slice still responded the same as they did in the living animal to the optogenetic light stimulation, but recordings could now be acquired from multiple locations more accurately. Here is an experiment on one rat that was trained on the low right task. The rat was sacrificed and the slice of its left striatum was recorded from a total of 14 locations. The darker dots are locations that showed synaptic strengthening. You can see that there are more dark dots on the low frequency side of the tonotopic axis. Plotting all of these 14 recordings from this one rat gets a summary statistic measuring the relative strength of the low versus high frequency synapses, the slope of this line. The authors did this experiment for 7 rats trained in the low right task and another 7 rats trained in the low left task. Data for all 14 rats is shown in this one plot. You can see that the black dots are mainly on one side of the striatal tonotopic axis for the low right rats and on the other side for the low left rats. Plotting each individual rat's synaptic strengthening gradient across this axis shows that the two different learning groups can be perfectly separated by this measure. Here is the how the authors summarize. This gradient correctly identifies learned associations in 14 out of 14 individual rats. The authors summarize their model of reinforcement learning with this picture. A naive, untrained rat has both high-frequency and low-frequency axons projecting from auditory cortex to both the left and the right striatum, and all paths are equally potentiated. When a rat is trained to associate low-frequency sounds with rightward licks, then particular synapses are strengthened in each side of the striatum. Now, this particular experiment was looking at the left striatum, which controls rightward licks. And the authors saw strengthening of these particular synapses, which formed the causal link of that learning. When the rat hears a low frequency, it is these synapses that make it more likely to choose the behavioral action of a rightward lick. The Zader lab that performed these experiments in 2015 have now replicated and extended these experiments, now using mice instead of rats. They did this in the recent 2021 paper that's shown here. Importantly, they showed that the stratum model that I just showed, that they proposed in their 2015 paper, is not quite the whole story. In this new paper, they trained 8 mice in the low left task and another 8 mice in the low right task. Just as in the 2015 paper, they found that they could distinguish these two groups perfectly based on the same type of acute slice measurement of the stratum. So, at one level, this is a complete replication of that original 2015 paper, but in mice. But this time, they also trained another set of mice with reversal. That is, first a mouse is trained in, let's say, the low left task till it achieves a high level of performance on that task. Then the rewards are reversed so that it looks like a low right task. The poor mouse is able to learn both the original task and the reversed, but perhaps surprisingly, the acute slices of these mice showed the pattern of learning associated only with the original learning, not with the reversal. This means that whatever new learning allowed the mice to learn this reverse task must have actually been encoded in a different brain area. Now, this may seem surprising, at least in light of the model that was just shown, but there is actually a long literature on learning and extinction and spontaneous recovery of learning that suggests that initial learning is never simply forgotten and overwritten. It's a fascinating literature that perhaps we can devote an upcoming journal club to. As we have covered in previous journal clubs, there are many other experiments that have shown that stronger synapses are also larger in their physical size as measured by light and electron microscopy. Therefore, it seems clear to me that the decoding of a learned behavior from post-mortem brain slices that was shown in this current paper could also have been done by other means than acute slice electrophysiology. For example, it could have likely been done by electron microscopy and connectomic reconstruction, of course at much greater effort. As such, this current paper adds evidence in favor of the hypothesis that learning and memory may potentially be decoded from a preserved brain using far future imaging technologies. Thank you for listening.